So Manus is a native of Galway, and he did a PhD at the University of Glasgow and a postdoc in Columbia University in New York, and has been a lecturer here and progressed his career since 2014. His research is in biomaterials and bioengineering, looking at the brain-machine interface and the development of electrically active nanotechnologies for regenerative medicine, biodegradable sensors, tissue engineering, mechanobiology. So Manus, I suppose, has been very successful in funding, securing funding, collaborations with industry, and also it's moving on me here. I haven't learned it off Manus. Has published widely. So I can see that we're also joined by Manus's family here tonight, so I think it's a lovely opportunity to have family come here, and because I'm sure you've been working very hard for all these years, and to see what is achieved, and again for all of us to see what's involved in reaching this level of promotion and the exciting research that's going on in the school that maybe we don't get an opportunity to see enough. So with that, I'll just hand over to Manus and welcome you to give your inaugural lecture.
Thanks. Thanks, Alicia. Thanks to everyone for coming. It's a real privilege to be here. It's super to be able to talk about my research, about my career and how I got here, particularly to my family, as Alicia mentioned. I have chosen quite a clickbaity title today. I'm going to explain exactly what I mean. A career in biomaterials and the importance of being promiscuous. As Alicia said, my name is Manus Biggs. I'm from the discipline of biomedical engineering, and I'm also a funded investigator with the Centre for Research in Medical Devices, or CURM. Right, what do I mean by this title? So my research focus for the past 15 years has been based on biomaterials. So essentially, I'm interested in developing new materials that can be used as either wearable technologies or implantable technologies. Biomaterials are interesting because they require very specific characteristics, very specific engineering needs to be involved in developing these materials, principally because the body is a very harsh environment. So they need to be able to persist within the body over chronic periods of time. Common examples are things like contact lenses, simple biomaterials, dental implants, catheters and stents, hip or knee replacements. These are all biomaterials, specific metals and polymers generally, that will persevere in the body and be able to carry out their function for decades, hopefully for decades. Where did my career start? I became interested in biomaterials, well, I started my career in Glasgow University, as Lucia mentioned. So I went to do a PhD under the guidance of Professor Matt Dolby. He is a bone biomaterials guy. His research career is focused on bone biomaterials. My research as a PhD student was funded by the AO Research Institute. So it's an institute based in Switzerland, and they research new orthopedic implants, hip implants, plates and screws, etc. So I went out to Switzerland for a while. Here I am in my 20s demonstrating why this institute persevered and developed in Switzerland. It was initially to help all the individuals that were breaking bones as they were skiing, which I enjoyed a lot. To give an example of the type of research I was doing in Switzerland and in Glasgow, if you look at this is a common implant, it's a hip implant. If you look at it up close, you'll see that it's quite rough. This is intentional. This isn't coincident with just manufacturing techniques. This is intentional. These implants are roughened, and this helps integration of the device with the surrounding bone. The stem is inserted into the medullary cavity of a long bow of the femur, for example, and it helps with the stabilization. What I wanted to do, or what my supervisor was interested in, and the focus of my PhD career, was to look at exact roughnesses. If we take what we were doing at the macro scale, micro scale, and then apply it to the nanoscale. If we create very, very small features, how do cells respond to that? Does that help integration of bone? Does it help cells adhere to these devices? Does it help with the integration and the stability and longevity, the chronic functionality of the device? Be it an implant, be it a hip knee implant, be it a plate or a screw, etc. And I really enjoyed this work. And so I thought this is what I was going to do forever. I was going to work in bone biomaterials, just like my supervisor. But I didn't. I moved to Columbia University in New York, and I went to work under another professor, Shalom Wind, and he was a specialist in a specific area, and it wasn't bone. He was a specialist in nanotechnologies and a specialist in immune biomaterials. He was interested in creating new immune biomaterials, biomaterials that can modulate the immune system. And specifically what he was interested in, if I can get this to work, is culturing T cells. So there's a great success story in science and engineering of CAR T cell therapies, taking patients' T cells, re-engineering them genetically in the lab, and then reintroduce them to a patient. And we do this, we modify these cells to be able to fight cancer more effectively. The way that we get these cells is we isolate them from the patient and we culture them in these great big bags, and it's expensive. And in these bags we have artificial antigen-presenting cells. You can see these bags here. And we have these antigen-presenting cells to prime the T cells, okay? So these are large bioreactors. You can see them here. What we wanted to do was look at if we modify the inner surface of these bags, can we more successfully prime the T cell populations? And so we created specialized biomaterials, immune modifying, immune modulating biomaterials. And we looked at the responses of T cells, and this was really interesting. And I thought I was going to stay in this field forever. I was going to be an immune biomaterial specialist, but I didn't. I moved from Columbia, and I came back to Ireland. Ireland is where I completed my undergrad degree. So I returned to Galway, and I went to conduct my first independent research career with a relatively modest grant from Research Ireland to look at neurospecific biomaterials. This is my first time I worked in neurobiomaterials. And I worked under the supervision of Abhay Pandit. Abhay Pandit is the director of CURM, the research center. He's quite promiscuous too. He'd be interested that I said that. Okay. There are also very good neurobiologists, neurologists in Galway, and they specifically look only at neural cells. They only look at neuropopulations. They're only interested in neurobiomaterials. And this is where I thought I was going to end up. I'm going to work in neurobiomaterials. This is going to be my field. Specifically, we were creating new biomaterials or looking at new biomaterials to increase the functionality of electrodes that are implanted into the brain. Can we use specialized materials to increase their function, to make them work better for longer, essentially? Here's an example of that. Here's an electrode, and we were modifying the tips of them with chemistries, with different types of physical modifications, using advanced biomaterials. Very interesting field, and I thought I was going to stay there forever. But I didn't. Where is my research focus now? So here, this is a relatively old photo of my lab. You can see I'm sans mustache. But these are some of the people that I've worked with. And to give you an idea as to where my career has gone, so here's our bone and immune biomaterials and brain biomaterials. I've augmented this career. Research in cardiac, lung, tendon biomaterials. Further still, cancer biomaterials, menopause-related technologies, drug delivery associated technologies, skin monitoring technologies. I'm going to talk about just two of these over the next few slides. So, to, I guess, maybe disappoint some of you, I'm not going to talk about how promiscuous I am in the typical or the common sense, which may be disappointing. But my wife is in the audience and I haven't been able to make eye contact with her, even though I did explain what I meant. But maybe we can put it to bed now. We can proceed and I can feel more comfortable. What do I mean when I say promiscuity and why it's important? I'm talking about the definition given here, Merriam-Webster. Promiscuous, adjective, not restricted to one class, sort, or person, indiscriminate. That is, my research interests are indiscriminate. Really, they're focused on what is the clinical challenge, what is the problem. And of course, we get these questions from physicians, and then it leads us to be able to, as biomedical engineers, try and address these challenges. So what is the clinical challenge? I'm going to talk about two, I like to think they're quite nice success stories. That's why I've chosen them, based on different biomaterials, based on different clinical applications. The first is going to be implantable bioelectronics. So this is an area that I've been very active in for the past 10 years or so. Bioelectronics have really captured the public imagination because they're quite interesting. They're thrilling to us. A lot of you have probably heard about the Neuralink device from Meta, not Meta, for Elon Musk's Neuralink, it's his company. Okay, so these are implanted technologies designed to either stimulate nervous system, the central nervous system in most cases, the brain, or to record electrical activity in the brain. So they stimulate, they send electrical pulses into, or they record, they receive electrical pulses. This is all accomplished via relatively simple technologies, battery packs, wires, simple microcontrollers. But principally, they all contain electrodes, penetrating electrodes. These electrodes are implanted into soft, squishy matter like the brain or the spinal cord or peripheral nerves with the intention of stimulating or recording. Here you can see this is a deep brain stimulation technology. This is one of the most successful bioelectronic devices. It's been clinically available for over 15 years now, maybe longer. These are devices used for the treatment of Parkinson's disease. So they're implanted into the deep brain region. I had a schematic up in a few slides previous. And they provide electrical pulses to be able to control some of the manifestations of degeneration, the essential tremor. So, what are the problems? What are the challenges with these technologies? The challenges are related to the foreign body response. So if you implant a device into the body, particularly something cold and hard like a noble metal, gold or platinum, or a polymer, silicon in some cases, the body rejects it because it recognizes that it's foreign, because it's mechanically dissimilar to the tissue that's implanted in. This is also greatly exasperated by the presence of micromotions. There was a video here that was showing micromotion of the brain. Okay, so you can see the brain is not anchored to the skull. It floats freely within the skull. It's like a jelly floating within a bowl. As you undergo locomotion, you walk around, as your heart beats, as you breathe, your brain slightly moves. And if you have a penetrating device, such as a sharp electrode stabbed into the brain, which is anchored to the skull, there's relative motion between the implant and the brain tissue. And this is problematic because it leads to chronic inflammation and it leads ultimately to device failure. So this type of challenge is why Elon Musk's Neuralink are not going to be commercial electronics anytime soon. You're not going to be fitted with them because this challenge is still unsolved. It's very difficult to solve this type of challenge. The challenge associated with the foreign body response to implanted neuroelectronics. Okay. So what happens? If you implant something sharp into the brain, particularly a metal, and these are conducting materials, so they need to be metal, or at the very least some sort of conducting polymer maybe, the cells become upset about it because they're soft, and these devices are hard and they're sharp. The support cells of the brain, the astrocytes and the microglia, will start to undergo degeneration. They become reactive. They'll start to become metabolically very active, and they'll start to produce pro-inflammatory cytokines. These pro-inflammatory cytokines establish a region of inflammation. And this is chronic inflammation. And chronic inflammation ultimately, over the course of years, leads to encapsulation. And encapsulation in this case means scar tissue deposition. So fibrous scar tissue is deposited by the astrocyte support cells of the brain, and there's also local cell death. So neurons die and the electrodes become encapsulated. If you encapsulate an electrode in an investing scar tissue, it's no longer effective at stimulating or recording because it's non-conducting. It's an insulating material that's wrapped around your electrode. This is problematic. It leads to increased impedance, so electrically speaking, it's not very stable. Reduced signal quality, you can't record neural activity anymore. The device fails. Neuronal loss and, of course, reduced device longevity. The device has to be replaced, it fails. So we knew that cells respond to this mechanical insult, and we wanted to know why. So we started looking at specialized ion channels. These are channels within the walls of the neurons and the walls of the support cells of the brain. And when they become mechanically stretched, they open up. These are called stretch-activated ion channels. They're responsible, for instance, for the perception of pain, of nociception. There are a whole load of these ion channels. They look something like this. As you stretch them, they open up, ions can flow into the cell, and these ions are secondary signaling molecules. They can cause specific cell functions, and we know that they're important in establishing inflammation. So we assayed for these, and we found that if we subjected cells to shear, we used a very simplified system where we can use mechanical shear via fluid flow, so we can culture cell populations, neural populations, onto a bioreactor system, flow media over them, and the cells become subjected to shear stress. This shear stress causes upregulation of the expression of multiple ion channels, and we found 2 were significantly upregulated, even at really low shear stresses. So these we found were they were responsible for some of the inflammation that was happening around the electrode. We then went on to show that this was in fact true in vivo. So we performed in vivo analysis with rats, where we have 2 control groups: our healthy rat brain tissue, a stab wound, which is just an electrode that's stabbed into the brain and it's removed and the brain is allowed to recover, or electrodes that are kept in situ for up to 8 weeks. And we found that, yes, of course, we expected to see astrocytes become angry and activated, microglia become activated, as we expected to see, and the neurons start to die. Okay? What we also found was when we looked at these ion channels, and specifically an ion channel called piezo1, this became highly expressed in the astrocytes that were around this glial scar. So we now had found a target that had become activated or upregulated in the support cells of the brain in response to electrode that's implanted chronically. What we wanted to do then was use a biomaterial. Can we use our expertise, or my expertise, in biomaterial design to create specific coatings for the electrode that's going to prevent this from happening? So what did we do? We looked to a platform technology that we can modify at the electrochemical and the biological level, and that's what we did. I'm going to talk about that specifically. And we've published widely on this biomaterial, as well as modified formulations looking at differences in physical properties. I'm going to talk about a biochemical approach, biochemical modification approach. So what did we do? This is what our electrodes look like under electron microscope. We took our electrodes and we coated them with a conductive polymer. So we're not using a hard material anymore, we're using a soft material because neurons prefer a soft environment. So we use a conductive polymer, a soft polymer, that we can use to coat the surface of our electrodes. But not just that, we also needed to graft a biochemistry into this polymer that was going to stabilize the environment, ensure that we don't have neuron death, ensure that we don't have activation of support cells. So what we did was we turned to a biochemist, a colleague of mine, and we asked it to create a heparin sulfate mimetic. So heparin sulfate is an element of the extracellular matrix. And it's present in brain tissue. It's very prevalent in brain tissue. And what it does is it acts as a scaffold for neurons to live within. And it also has an important function in binding growth factors. So it sequesters growth factors to the extracellular matrix of neural tissues. Okay? So we took this heparin sulfate and we made an analog of it, a synthetic analog. We didn't use heparin sulfate. Why? Heparin sulfate itself is very unstable. It undergoes enzymatic degradation or decay quite rapidly endogenously. It also doesn't bind to very many growth factors. So what we wanted to do is to make that heparin sulfate mimetic stable and to make it have high affinity for growth factors. So what we did is we engineered it. We called it F6. F6 is a heparin sulfate mimetic that's resistant to enzymatic degradation, and it also contains lots of sulfate groups, and these sulfate groups are important for binding of growth factors. So in effect, what do we have? A conductive biochemistry that we coat the surface of our electrode with, and this will present heparin sulfate. The heparin sulfate will bind endogenous growth factors. So we're not relying on supplying growth factors by coating our electrode in growth factors, which has been sort of looked at for a long time. It doesn't work very well. We instead coat our electrode with a sequestering chemistry that will bind endogenous growth factors and present them to the neurons. Okay? So what do we do? We conducted a plethora of in vitro studies. We looked at if we create coatings on our electrodes and we culture neural populations on it, what do we see? Well, initially we saw a really nice reduction in pro-inflammatory cytokines. So the cells aren't becoming inflamed anymore. This is all performed in vitro. And we used mixed population of rat neural cells. We found significant reduction in pro-inflammatory cytokines, interleukin-5, interleukin-6, TNF-alpha, INF-gamma. So this is encouraging. We then performed really nice with colleagues of mine, both in the University of Paris and here, in the front row. We performed binding analysis. So we wanted to prove that, yeah, our growth factors actually are being sequestered to our chemistry. The F6 heparin mimetic is binding growth factors. So we performed competitive binding analysis. This is done in a wet state. This is done in the lab. We add different concentrations of different growth factors into our F6. And we see that we get this very high binding affinity, very high binding rate, even at very low concentrations. This is really encouraging. It means that our mimetic actively binds growth factors. We studied fibroblast growth factor, VEGF, and glial-derived growth factor. These are all really important in neuroregeneration. So these are growth factors that promote regeneration of neural tissues. We also did some really nice computational work. So we modeled this computation using molecular dynamics. So you can see, it's not playing. Never mind. Sorry. No. Never mind. We'll move on. Okay. And finally, then we wanted to have a look at what was the response in vivo. So we took our electrodes, and in the same model that I presented, we coated electrodes and we implanted them into the brain of rats. And this was an 8-week study. And after 8 weeks, we assessed what do the support cells look like, the astrocytes and the microglia. Are they still inflamed? Do we still have this scar tissue forming? What do the neurons look like? We were a little bit disappointed initially because we only saw a really moderate attenuation of gliosis. So gliosis is the fancy term for scar tissue deposition in the brain. We only saw that if we look at, do a comparison between uncoated platinum electrodes, these are just the basic metal electrodes. If we look at just the conducting polymer and compare it to the conducting polymer with the mimetic, with the biochemistry, we only see that we have a reduction relative to the uncoated. But the conducting polymer alone, as well as the conducting polymer functionalized materials, acted in relatively similar way. Moderate reductions in gliosis. We saw the same thing with microglia, the immune cells of the brain. Moderate reduction. What was really nice is when we looked at the presence of neurons. So if you look here, in fact, I can zoom in on it. These red dots are individual neurons. This gap here is where the electrode was. And you can see that this is just the conducting polymer. This is the conducting polymer plus our heparin sulfate mimetic. You can see that there's far higher density of neurons. So what we can claim is that the heparin sulfate mimetic is neuroprotective, it's neurotropic. It's keeping those cells alive around the electrode that ordinarily would die. This is really important. This has significant implications for the functionality of the electrode. And of course, we performed physiological recording, so we use these electrodes to record neurons firing, and we found that these electrodes were significantly better at capturing electrical activity of the brain because the neurons aren't undergoing death around the electrode. So this was really encouraging for us. What are we doing next in this work? We are hopefully moving to clinical translation. This is in conjunction with a PhD student of mine. Now, we're not going straight to the clinic with implantable electrodes into the brain. We're using a sort of a more accessible, low-hanging fruit. We're functionalizing little microneedles, and we're using these for monitoring skin. So same type of technology, but it's implanted onto the surface of the skin. Why do we do this? Because we want to help surgeons that are performing skin grafting techniques. Skin grafting is associated with a relatively high failure rate, can be up to 10%. And this all happens within the first 48 hours. So after transplant of the skin tissue, the transplant needs to be monitored really carefully for about 48 hours. After that, you're out of the danger zone. But within those 48 hours, there's a likelihood that the perfusion, the blood perfusion into the skin can become disrupted and the skin will die. So what we are proposing to do is to take our microelectrodes, you place them on the skin, and they record the blood flow in the tissue. And they do this for 48 hours, and then you don't need to use the device anymore. We're going to coat it with our technology, and hopefully this will increase the efficacy, the recording efficacy of these technologies. And of course, this is linked to a microcontroller, and this will alert a physician if the blood flow is disrupted in skin tissue. So this is where we're hoping to move. We've applied for funding to carry out a preclinical trial using this type of technology. How am I for time?
You're fine, you've got 10, 15 minutes.
Great. Okay. So I'm going to draw a line on that story, and I'm going to quickly move on to another one. Hopefully, it's only going to be, 5 minutes, no problem. I did. We just at the intermission, so it's all right. Okay, I'm going to talk about another technology, again, just to demonstrate, the different areas that my research has sort of brought me to. I'm going to talk about a technology that we sort of developed in conjunction with a industry partner. So a lot of the research that we conduct within the department and within the research centers I'm associated with are working with industry partners. And this is with an industry partner called Intuitive Technologies. They're based in the US, and they create orthopedic devices. What they wanted to do was to create a tendon anchor, an improved tendon anchor. Every year, there's about 6 or 7 million tendon injuries. And the way that these are treated by a surgeon is via a tendon anchoring approach. A lot of the tendon ruptures clinically are not that the tendon is snapped in half, it's that the tendon is removed from the bone. I assume it's really painful. It sounds painful. But it's not that the tendon is snapped per se, but it's wrenched from the bone. The way that these are treated is with an anchor, and it's just it's like a dry plug in a wall. And a surgeon will come and he'll create a drilled hole. Into this void, he will place this anchor, and the anchor will be sutured to the end of the tendon that's been removed from the bone. These tendon anchors are typically PEEK, which is a polymer. It's a non-resorbable, really robust synthetic polymer, or titanium. So relatively tough materials. There are problems with these, however. Suture anchors are the gold standard for tissue-to-bone fixation. The common mode of failure occurs at the suture-tendon interface. So they're associated, again, with a relatively high failure rate. And the failure occurs as the tendon slips from the suture, or the anchor is pulled from the void. Essentially, it'll wiggle a little bit, and then eventually it will slip from the void that's been drilled by the surgeon. And this can occur relatively quickly, or over 2-3 years after this has been fixed. Okay, so what do we want to do? We wanted to create a better tendon anchor, essentially. So we turned to a biomaterial that had been published many years before. And this is really nice for several reasons. We made a composite material out of citric acid. Citric acid is what makes lemons nice and tangy and tart. We created a polymer out of this material. It's called polyoctamethane citrate. And we took that and we mixed it with hydroxyapatite powder. Hydroxyapatite is a mineral found in bone tissue. It's a microscale powder, so a very fine powder. We mixed it with our polymer, our lemon polymer, citrate polymer, and we created a really stiff, bone-like material. What's nice about this material is that, well, a couple of things. One, it resorbs over months and years, so it will slowly degrade and it will slowly be fully resorbed. It'll degrade. The other thing is that citrate is really important in bone metabolism. Citrate is important in bone growth. So the fact that we have made our device out of citrate is a really nice approach. Another thing about a material is it contains our hydroxyapatite ceramic powder. Hydroxyapatite is important in mineralization. Hydroxyapatite, that ceramic, is osteoinductive. It causes mesenchymal stem cells in bone tissue to differentiate into bone-like cells. So we can say that we have a material that's going to aid in the deposition of new bone, and it's going to induce new bone formation. And it's going to be resorbable. We chose this material, and we advanced it further. We wanted to look at the cell responses in vitro and in vivo. Here is a promotional video where you can see what I'm talking about. This is the anchor. There's no suture with it, and you'll see why. So there's the void drilled by the surgeon. The tendon is placed into the void, and then this screw is rammed in, and the tendon is pinned up against the wall of the void. There's no suture per se. The anchor will slowly degrade over the course of years, and the tendon remains in place. Here you can see that. Day one, surgeon creates a void. The tendon is placed in there, and the citralock is the tendon anchor. Over the course of about 12 to 24 months, the material will start to break down. So the citrate and the calcium and phosphate ions from the ceramic will start to leach into the surrounding tissue, into the void. That leaching of materials will induce new bone deposition. And as the device degrades, new bone fills that void until eventually in the late stage, the void is completely absent and the tendon is trapped within that void and the device is completely resorbed. There's no more any traces of it. Is the theory. Okay. So what did we do? Oops. We wanted to look at several things in vitro. Basic things, first of all. This is what we were tasked to do. How quickly does the device degrade? And we wanted to do this with a couple of different formulations. So we studied two different formulations of our material. One was, we call POC 1.1. This has a 1-to-1 ratio of the polymer to the ceramic. And another was POC 1:1.3. So it had slightly more ceramic. We looked at these two formulations. And I refer to these as POC 1.1 and 1.3 going forward. Okay? One is slightly more ceramic. The other thing we looked at was a gold standard material. These materials, PLA, TCP, these are used clinically. These are used commercially in orthopedic bones and plates. So there are other companies that make these resorbable devices. Not tendon anchors per se, but resorbable screws, for example. These are made from PLA, TCP. Sort of a similar chemistry, but without the citrate. Okay? So we compare all our results to that gold standard. What do we find? That over the course of, we performed an accelerated aging study, over the course of 120 days, which equates to about 3 years, physiologically speaking, we have significantly slower degradation of our technologies relative to clinical gold standards. So we engineered our materials to degrade at a far slower rate. They also had a far smoother surface profile because they weren't degrading as fast as the PLA materials. We performed mechanical analysis. Mechanically speaking, our materials were far lower elastic modulus. They were far more robust mechanically. They were stronger and less brittle. So the competitors' industry gold standard are harder, but they're quite brittle. Our materials are slightly more elastic, more robust. They had a strain at peak stress of about 50%. Relative to 5%, so order of magnitude greater than the gold standard, and they had significant increase in their peak stress, about 240, 220 megapascals relative to the gold standard. Again, we performed a whole slew of in vitro work. Specifically, we want to look at, well, if we take stem cells, mesenchymal stem cells, and culture them on our materials, do these stem cells start to did they become induced to differentiate into bone-like cells? This is really important for regeneration in bone tissue. And what we found was, and there's a lot of data here, but just look at these blue bars and the red bars. Okay, these are our 1.1 and 1.3 materials. And the green bars are our gold standard material. And what we see is that by day 21 of culture, we have significant increase in the expression of these mineralization-associated proteins. So osteopontin, excuse me, deposition of calcium, and the production of alkaline phosphatase. So these are things that are important. They're indicative, they're markers of differentiation into bone cells. So what we could say is that the MSCs cultured on these materials were starting to become more bony-like, which is exactly what we wanted. If we can induce differentiation of mesenchymal stem cells into bone cells in vitro, then that's indicative that this is a process that will happen in vivo, that the cells, the mesenchymal stem cells that line the surface of bone can start to differentiate into bone-like cells and deposit new matrix, which is exactly what we wanted to see. We then completed a really extensive in-depth analysis of proteomics. So we wanted to look at the transcriptome of these cells at a far greater level, and we performed what's called RNA sequencing. So we looked at hundreds and hundreds of different sequencing events, different transcription events, and we were able to look at taking hundreds of genes and grouping them into different pathways. So we can look at what happens at the pathway level. When we did this, we were able to quantify changes in things like osteoinduction, osteoconduction, growth factor signaling. These are events really important in bone regeneration. What we found was, if we look at our POC is to 1.3 material, this is our nicest composite that performed really well. significant upregulations in osteoinduction, but also really encouraging in the deposition of mineralization and the calcification of the extracellular matrix. Again, it's exactly what we wanted to see. At the transcriptome level, these MSCs were starting to induce processes that were indicative of the deposition of mineralized matrix, or bone tissue, essentially. So we were encouraged by that, and we then went to perform an in vivo study. So in a sheep model, we looked at initiating a preclinical trial. So this is low-bearing tissues. So we, again, just as we saw with the surgery in the informational video, surgeon created a defect into the sheep tibia. The tendon was placed in, as well as our biomaterials, and we looked at the response over time. Now, in hindsight, this study should have gone on for longer. 6 months was inadequate, and we only advanced some of the groups to 12 months. But our citrate, and we only advanced one formulation of our citrate polymer, and we compared it to the gold standard. What we found was that it performed equally or better than up to 6 months in vivo within the animal. But what's really nice is that after 6 months, the pull-out strength was significantly higher for our material. So we performed an assay where we look at how much force is required to remove the tendon from the bone. And what we found is significant increases in the forces required to pull out our citrate tendon. And this is exactly what we wanted, even though ideally this study would have run for 36 months. This is a very tantalizing early result though, and it was on the back of this result that this device was taken to clinical trials and it performed very well. So just to give you an idea as to what happened in the timeline for this technology, in 2016, I first met the team from Actuative. They met with a team from Cura, me being one of them. The study, our joint enterprise, our study started together about 2018. In 2020, they received FDA approval for Citralock, which is the device I've been talking about. In 2021, they received FDA approval for Citraflex, a related product, and in 21, another related product. This is what the video was sort of based on here, this Citraspine technology. And then in 2021, this technology was licensed by Stryker and it's now clinically available. So this research was translated clinically into a really successful product that's being used clinically. It was really encouraging. It's an example of a really nice success story to be involved in as an academic, where you can see it's sort of the fruit of your labors being translated clinically, particularly working in the Department of Biomedical Engineering. It's really refreshing. It's really encouraging to see that type of translation. Anyway, I'd like to finish just by mentioning I've been very lucky to be able to publish all these studies. Again, it's really encouraging to see these types of studies being published. You know, when these land on your desk or you see them being published online, it's really encouraging. I've been very lucky to be involved in a number of studies. Again, this has really helped in my career. Being able to publish these studies has really helped me to get where I am today. As well as that, it's been really enjoyable to be able to interact with the public and with patient groups, to be able to talk to people about science, to be able to talk to the public about science. And I've been very lucky to be able to do that. I've spoken on radio and television talking about my research, and I've got good feedback because public likes to know what's going on in universities. They like to know what new technologies are being developed. Particularly patients want to know, you know, what's going to happen next. Where is this? Where are the therapies coming from? Where are they going? So it's been a really rewarding aspect, I guess, of my career, being able to talk to the public. And finally, I'd like to thank everyone that's been instrumental in my career for the past 10, 15 years. I've certainly left out many people from this list. Don't be offended if you're not here. I have done my best, but I'd like to say thanks to all researchers and colleagues from the University of Galway, UCD, University of Paris, particularly the researchers that were involved in the studies that I just presented today. This was funded by Research Ireland. A lot of it was funded by Research Ireland, as well as the European Innovation Council. They've been instrumental in allowing me to conduct these studies and for my career progression. So I'd like to thank everyone for attending. Thanks so much for coming. Please ask me questions if you feel we have time for it.
Questions. First of all, again, massive congratulations, and it's wonderful to see your two stories. I'm aware that you have lots of other interests, and Biomaterials is the nexus.
I picked the best.
Yeah. It was nice to see those two stories. I might ask a quick question, and if others have questions, I'm just wondering about your electrode work. You focused a lot on the mechanical changes that might be activating these glial cells. But what about thermal effects? Because you see a very similar effect with when you have very small increases in temperature in terms of apoptosis of cells and proliferation of cells and that kind of stuff. So is there any thermal effects?
There aren't any thermal effects with recording, obviously not, which the study was sort of looking at. With stimulation, there can be, but generally the pulse width and the pulse duration is designed to avoid thermal effects, because if you've ever worked with neurons, they're exquisitely sensitive to everything. They're very difficult to culture because they like to die. If they're shocked when you're taking them out of the culture environment, they die. If you accidentally leave them out for too long, they die. Any thermal effects is going to significantly cause issues. So it's important that they're looked at.
So in your specific experiments, you don't think that there was thermal effects involved? And did the coating do anything to modulate that?
Not the coating per se, doesn't act as a heat sink or anything like that, but the pulse width and duration is optimized to prevent thermal effects. But there certainly are thermal effects with other neuromodulation approaches, particularly using ultrasound. And that's something that we're actively looking at right now. It's very easy to raise the temperature using ultrasound. So it's very non-invasive, but there's also that issue with thermal effects.
Okay, great. Do we have any other questions for Manus yet? Pat?
Hi, Pat.
Great talk, Manus, and congratulations. I think it's wonderful the way you've demonstrated, the vast array of areas that biomedical engineers can work in. They're adventurous, or promiscuous, as you say, and just shows the huge range of scales at which we have to work. So thanks again. It was fantastic. Just a technical question, just in terms of the degradation of the anchor. It must be a very difficult engineering challenge to tune the degradation versus the bone ingrowth, particularly for something that's massively low bearing as a tendon anchor.
Yeah.
Is there further work to do there?
So those iterations that I mentioned in the sort of success timeline, they were all different iterations of pretty much the same material. The only difference is really the cross-linking density of the polymer, and specifically looking at that varying in the degradation time. So it's really important depending on the clinical application. You saw the last one that was developed 2021-22. That was specifically looking at spinal injuries, so fixation of the spine. And that was all the research effort. I wasn't involved in the development of that device per se, was looking at modulating the degradation rate for that.
Great. Okay, I'm looking at, we're coming up bang on at 7:30, so that was the time we'd hoped to finish. So first of all, I'd like again to give a round of applause for Manus, and congratulations on your promotion. So with that, I'll just wrap up. So we have some food, some finger food and stuff outside. So please stay around. You may have lots more questions for both of our new professors. So please, I'm sure they'd be delighted to talk more about it. I'd like to thank Magda again for organising all this, and Dave Finn as well. Is Dave here? He's outside, and we've obviously got our support from Cormac here for the audiovisual. So thanks to everyone for coming as well. This is going to continue. Hopefully each year we'll have a number of successful promotions, and we'll have the opportunity to come together and learn about their fantastic research. So please make sure to stay engaged, and again, thanks everyone for coming. Great.